Hi and welcome to this Microbiology Basics video on bacteriophage replication, where I'll briefly recap on what a bacteriophage actually is. I'll then describe the various components that make up a bacteriophage, and then I will go on to detail the bacteriophage replication process through an animation that focuses on both the lysogenic and lytic cycles. So hopefully after watching this video, any questions you had before you clicked on the video will have been answered and you'll be more informed going forward. So with that, let's crack on and I hope you enjoy the video. So before we go through the ins and outs of the different parts of bacteriophage replication, there are some key things we need to know about bacteriophage. The first is that they are obligate intracellular viruses that specifically infect bacterial cells. Basically, they are viruses for bacteria. A second key point we need to remember is that just like all other viruses, they are metabolically inert, which is just a fancy way of saying they are unable to do anything by themselves because they don't contain the enzymes required to transcribe and translate their genomes. Therefore, in order to complete these fundamental processes, they need to hijack the enzymes of a host bacterial cell. And this is exactly what you are seeing in the picture on the right. Multiple bacteriophage are attaching to and invading a bacterial cell in order to utilise the enzymes within. So before we move on and look at how bacteriophage replicate, let's look at their structure, which as you can see from the pictures taken with an electron microscope is highly complex. So on this slide, we're going to particularly look at the structure of bacteriophage T4, which forms part of the Myviridae virus family, which are capable of infecting Escherichia coli. If we start at the top with the most important aspect of the bacteriophage, we have the capsid. This is a proteinaceous structure that contains the bacteriophage genome, which contains all of the genes required for the production and dispersal of progeny bacteriophage. Next we have the collar. This connects to the capsid and aids environmental sensing as well as the assembly of progeny virions once inside a host bacterial cell. Attached to the collar is the sheath structure. This is a long helical hollow tube structure that allows the viral genome to pass from the capsid into the host bacterial cell through a complex contraction and injection process using its core injector that I'll go over later. Attached to the bottom of the sheath is the base plate. This structure senses the target site of the host bacterial cell and mediates the contraction and injection of the viral genome. And finally, we have the tail fibres. These are responsible for the initial sensing and binding to specific anchor points on the host bacterial cell. Now when we talk about the replication process later, I'll recap on what I've just said so that you can visualise what actually happens when a bacteriophage approaches and attaches to a host bacterial cell. But before we do that, let's go back to the structure of our T4 bacteriophage, which is essentially comprised of three major components. We have the head, that contains the genome, the tail, which contains the mechanism to inject the genome into the host bacterial cell, and the fibres, which mediate the recognition and binding to a specific host bacterial cell, which in the case of bacteriophage T4 is Escherichia coli. Now as with other viruses, bacteriophage come in a range of different shapes, sizes and complexities, and whilst bacteriophage T4 is the most common depiction of a bacteriophage in textbooks, most likely due to its overly complex shape, feature set and futuristic looks, there are many different varieties. For instance, bacteriophage lambda, which also infects Escherichia coli, is slightly less complex with fewer tail fibres and a flexible tail. And we also have bacteriophage M13, which again infects Escherichia coli and is completely different, with a helical filamentous shape. Additionally, some look very similar but infect different bacterial species, so bacteriophage SP01 for instance looks very similar to bacteriophage T4 but it infects Bacillus subtilis. Now as I've just mentioned, each phage tends to be specific to a certain bacterial species. However, multiple different phages can target the same bacterial species. So for an example, Escherichia coli is susceptible to infection by bacteriophages lambda, T4, M13, P1 and mu, whereas each of these bacteriophage can only target Escherichia coli. Okay, so that's the basics out of the way. Now we'll move on and look at how bacteriophage replicate, which is obviously a complex process because, as I said earlier, they don't contain any of the necessary machinery to do this on their own. They only contain the genetic code for it all, and so the entire process starts with the hijacking of a host cell. So here we have bacteriophage T4 in the environment along with our target bacterial cell, which in this case would be Escherichia coli. Now, I have only shown a single T4 bacteriophage here, however it's highly likely that there will be multiple copies of the same bacteriophage within the environment to help speed up this initial process whereby the bacteriophage comes in close proximity to the bacterial cell surface. If we zoom in, we can see the various components of the T4 bacteriophage, and to just quickly recap, the capsid contains the viral genome, the collar attaches the capsid to the sheath, which is used to inject the viral genome into the bacterial cell, once the tail fibres and base plate have irreversibly bound to the receptors on the cell surface. Now, the receptors on the surface of bacterial cells can come in a range of different shapes and sizes. Here I've only shown a very basic lock and key representation of the proteins. However, the target sites are cell surface proteins that provide critical function to the bacterial cell and are therefore highly unlikely to change over time. Examples of common cell surface receptor proteins include lipopolysaccharide components, outer membrane protein complexes, and even flagella proteins. So as the bacteriophage approaches the surface of the bacterial cell, the tail fibres start to sense and identify key cell receptors that it can bind to. More often than not, it will bumble across the surface until the tail fibres bind irreversibly to their chosen cell receptor. Once this happens, the bacteriophage is lowered onto the surface of the cell so that the base plate aligns itself with its own receptors. This signals to the sheath of the bacteriophage to contract, and in doing so it crumples down on itself, injecting the core of the sheath into the bacterial cytoplasm. The viral genome of the bacteriophage is then injected into the host bacterial cell cytoplasm at a rate of around 3,000 nucleotides per second, which is considerably faster than other methods of inserting DNA into a bacterial cell, such as conjugation and transformation, both of which only occur at a rate of 100 nucleotides per second. Now, once the viral genome is within the bacterial cytoplasm, it circularizes in order to protect itself from host bacterial nucleases. To achieve a fully circularized genome, it uses host DNA ligase enzymes to reanneal the ends of its viral genome. Following the circularization of the phage genome, it then makes its way to a specific point of the host cell chromosome, where it integrates itself into the genome via site-specific recombination using virally encoded recombinase enzymes. Once the bacteriophage genome is fully integrated into the host chromosome, it is known as a prophage, and the bacterial cell itself is known as a lysogen. Now, because the prophage forms part of the bacterial genome, with each cell division cycle it gets duplicated along with the chromosome so that when the cell divides, each of the two daughter cells contain a copy of the prophage. This is then repeated indefinitely as a bacterial population grows and is known as a lysogenic bacteriophage cycle, whereby the phage genome lies in a state of dormancy. During this dormancy, expression of phage genes is repressed within the lysogen by specific repressor proteins. In addition to this, these repressor proteins stop bacteriophage superinfection, which is where the same bacteriophage tries to infect a cell that already contains the same prophage. Now, that isn't the end of the road for our bacteriophage, and it can quite easily come out of its dormant prophage state and enter what is termed a lytic cycle. The lytic cycle usually starts when the host bacterial cell containing a prophage is put under external stress that damages the DNA, such as ultraviolet light. This causes the prophage to pop out of the bacterial chromosome, and through multiple transcription and translation events using the host bacterial enzymes, the cell unwittingly produces all of the components needed to make bacteriophage progeny, which are assembled and amassed within the cytoplasm. In addition to the production of progeny virions, virally encoded lysins are also produced, which accumulate within the cytoplasm. Once a set number of bacteriophage have accumulated within the cell, known as the burst size, and the concentration of virally encoded lysins hits a threshold, the bacterial cell is lysed open, releasing the bacteriophage into the environment so that they can go on and infect other bacterial cells. This completes the lytic cycle of the bacteriophage, which is considerably different from the dormant lysogenic cycle. So to quickly summarise and recap, the replication process starts with the bacteriophage sensing and attaching to specific receptor proteins on the target bacterial species. Once the bacteriophage has bound to its target host, it inserts its viral genome into the cytoplasm of the host bacterial cell. This is circularized and incorporated into the host cell genome through site-specific recombination, creating a lysogen cell that contains the prophage. The production of repressor proteins within the lysogen allows the prophage to enter a state of dormancy whereby it is passed on to daughter cells through the bacterial cell's natural cell division process. This is known as the lysogenic cycle. Now at some point the bacteriophage will enter a lytic cycle. This usually occurs when an external stress is placed on the bacterial cell. Essentially, the production of repressor proteins stops and the prophage is excised from the host chromosome. Using the host cell machinery, the viral genome is transcribed and translated in order to produce progeny virions along with cell lysis enzymes, and once a set concentration of both of these things occur within the cytoplasm, the bacterial cell is lysed open, and the bacteriophage exit into the environment to start the process over again. So that's the basics of bacteriophage replication. I hope it answered any of the questions that you had at the start of the video. If not, you can ask me questions on Twitter or the MicroChat Discord server, links of which are in the description, or you can comment on the video down below. If there are any video topics that you'd like to see in the future within the biological sciences, then please let me know. Be sure to subscribe for more videos like this, and hopefully I'll see you in the next one.